Good day, grade 12 learners. Welcome to today's Life Sciences lesson. My name is Vedika Maharaj and I'm one of the subject advisors for Gauteng. This lesson is brought to you by Gauteng Department of Education in collaboration with Sibona Discovery Center and broadcasted by Bakama Research and Development. Today we'll be teaching you about genetics and inheritance, specifically concepts of inheritance, monohybrid crosses, and sex determination. My partner will be Nonhlanhla Mshali, and she will be joining us later on in this lesson. So, where does genetics fit in? Where does this topic fit into our syllabus? It starts at the beginning of term 2 in grade 12, and it forms part of paper 2. It is 48 marks of paper 2, which means it's about a third of your paper 2, which is quite a large chunk. So it's a very important topic that you need to make sure that you understand, and we're going to take you through it today. As I explained, we're going to be doing concepts of inheritance, monohybrid cross, sex-linked inheritance, and the other three, pedigree diagrams, genetic engineering, and paternity testing, will be done on a different day, in a different lesson. So let's just introduce genetics to you a little bit. What do we understand when we say genetics? When we're talking about genetics, we are talking about the study studying the principles of heredity and variation. What do we mean by heredity? This is the passing of characteristics from parent to offspring. So let me give you a little bit of background. You know, when you start in term 1, you would have done DNA. Well, the first time you actually would have heard about DNA was when you, in grade 10, when you studied mitosis. Right? You studied about DNA and you studied mitosis and you studied chromosomes. So this DNA information is actually going to be carrying our genetic information, right? So in grade 12, in term 1, you studied DNA, the code of life, and there you actually studied how DNA has the code for protein synthesis. Okay, so in the same way, DNA also has the code for our genes or our genetic makeup, our characteristics that are passed down from our parents to the offspring. And this DNA ensures that the offspring resemble the parents and also contributes towards genetic variation, which in turn assists with survival of the fittest, which you will learn more about in term 3 when we do evolution. In term 1 in grade 12, we also learned about meiosis. During meiosis, at the end of meiosis, we have the formation of gametes, haploid gametes. In the female, the haploid gamete is an ovum, and in the male, it is a sperm cell. During reproduction, the ovum and the sperm fuse, and during fertilization, and results in a diploid zygote. That diploid zygote is going to have one set of chromosomes from the father and one set of chromosomes from the mother. So in actual fact, because the DNA is carried on the chromosome, that will mean that that zygote will have half the DNA from the mother and half the DNA from the father. That is the whole crux of genetics and this of heredity. Sorry. The next slide talks to you about the concepts of genetics. But before I mention that, I just want to mention to you that there is a man called Gregor Mendel. He was an Austrian monk, and he actually did some experiments. He was named the father of genetics, and he did some experiments and observations on garden pea plants. And this actually contributes to our understanding of genes and how they are passed from parents to offspring. Now, at the time when he did these investigations and these observations, he did not know that they were called genes at that time. He just realized that they were factors that were present in the parent and the offspring. So these are the concepts of genetics. There are terms that you are expected to know. Please know them well. They are commonly confused and they are also used interchangeably. The first one is chromatin versus chromosome. So the chromatin is over here. It is found in the nucleus of a cell, and they are thread-like structures which carry DNA. And if you can see the way it is actually represented here to show you that the DNA double helix is wound around the histones, and later on when they become a chromosome, then you will see that it's still a thread-like structure, but it becomes more condensed, and it becomes more compact, you could say, and it carries the DNA or the genetic information. Genes versus alleles. A gene is a segment of DNA or chromosome that codes for a particular characteristic. An example of this is, let's say this one, it is the color of the flower, right? And an allele is the alternate forms of a gene localized on the same locus on an homologous chromosome. So when we talk about locus, we are talking about the position of the gene. So an example of this, of an allele, would be purple or white. So the color is the gene, right? Or the trait or the characteristic. It's the gene. It's the segment of the DNA which is going to code for the particular characteristic or for the particular trait. So in this instance, the example is the color of the flower. Whereas the allele is the gene is found, that gene for color, for flower color, is found at a specific point on the chromosome. This is the point in this particular diagram where they're showing the position, which is the locus. And what we mean by alternate forms is that this one is the color. It can either be purple or white in this instance. So on this chromosome at that position, it is coding for purple, and on this position, it is coding for white. Then we have phenotype and genotype. The phenotype is the external physical appearance of an organism. The phenotype is determined by the genotype. So it's what you look like on the outside. If you look at this diagram that I've inserted here at the bottom, you will see that you can physically see the different characteristics of the mouse, of the flowers, the different color flowers, and the different insects that are here. This is what you physically see, the physical appearance. Whereas the genotype refers to the genetic composition. Okay, so I would remember it as the PH over here, standing for physical, and the GEN over here, referring to the genetic composition of an organism. And another thing that's important about the genotype is that it is represented by letters. Okay, as they have the example here, they are represented by letters, and those two letters represent an allele. So this capital B here is representing an allele for a specific characteristic, and this one over here, well, in this case it's the same. They're representing the same characteristic, but the main point to understand for now is that the genotype is represented by letters which are representing the two different or the same alleles. Homozygous versus heterozygous. If alleles of the same characteristic are both the same, the organism will be homozygous for that characteristic. If the alleles for a characteristic are different, the organism is described as heterozygous for that characteristic. Okay, homo also referring to the same. So let's have a look at this picture. The picture will help us understand it better. So if we're looking at this, we're looking at homologous chromosomes as indicated here in the first diagram. We have homologous chromosomes in a diploid organism. Okay, and then we have the two genes. The gene is here. Remember I said this gene is coding for a particular characteristic. So it will be at the same place or at the locus on the homologous chromosomes. And in this case, we have the one allele over here, which is the capital B, and we have the other allele over here, which is another capital B. Because they are the same, they are called homozygous. In this diagram, it's also a diploid organism. It's important to note that it's the diploid organism because it's carrying the full set of chromosomes. Okay? diploid, which is the full set of chromosomes. And here we have at the same, once again, you can see that it's at the same position on the chromosome, so that is the gene, and then the one allele is capital B, the other allele is a small little b, and because they are different, they're coding for the same gene, but they are different, they are heterozygous. So one is maybe, as we mentioned before, purple, and the other one white. In this, that would be heterozygous. In the last diagram, they are showing you that we have, again, the same, I'm reiterating the same point, that we have the gene at the same position or at the locus on the homologous chromosome. In this instance, we have a small letter b and a small letter b. They are both the same, so they are also termed homozygous. Okay, so the two homozygous conditions you can get are either with the capital letters or the small letters, which I'll explain to you in more detail in the next slide. Or you can have the heterozygous condition, which would be different alleles. Right, then we come to the dominant versus the recessive allele. The dominant allele is the allele that masks or suppresses the expression of the allele partner on the chromosome pair, and the dominant characteristic is seen in the homozygous and the heterozygous state. Okay, so that's a mouthful. So let's quickly explain it better. So it's going to mask, or it's going to suppress, or it's going to basically override, okay, what that other allele is going to be expressing. So the dominant one, it's like it's taking charge, and it's going to be putting the other one aside, and therefore it is going to be expressed in the phenotype. So even though the genetic makeup is showing that there are maybe different types of two differing alleles, the dominant one will be the one that is expressed in the phenotype, in the physical appearance. Whereas the recessive allele is the one that is suppressed. It's the, and the recessive trait can also be expressed in the phenotype, but it will only be expressed when it is homozygous recessive, if both the alleles for the trait are homozygous recessive. Okay, so there's a nice, lovely diagram over here that's going to explain everything that I basically said now into more, just to round up everything. Okay, so here we have a P, okay? Let's look at the phenotype first. The phenotype is showing us that it is smooth, okay? And this one's phenotype is showing us that it is, round, not smooth, round. And this one is showing us that it is wrinkled, okay? This is what we can physically see. So in this one, we can physically see that it is wrinkled, and in this one, we can physically see that it is round. Now we're looking at the genotype. Remember I mentioned that the phenotype is going to be determined by its genotype. So here's the genotype. It is the genetic composition or the genetic makeup, and the gene is here on the same. You can see it's at the same point. Okay, the gene is here at the same point, but one gene is, the one trait over here is being expressed as wrinkled and recessive. And this one is being expressed as round and dominant. So it's indicating to us that the dominant one is round. Okay, and to actually explain to you further, the dominant is always written with a capital letter. So capital letter is dominant, and the small letter is recessive. Right, so in this instance, they're telling us now that the round is the characteristic which is dominant, and in this instance, it is wrinkled, which is recessive. So we have one capital letter and one small letter. So as we mentioned previously, this would be heterozygous, right? And then, so they have contrasting alleles. That is the heterozygous condition. And it is the reason why it is round is because the dominant allele here for round is masking or suppressing the recessive allele. In this diagram, we have the phenotype once again, and then which is going to be relies on what is the genotype is. And here we have that it's wrinkled, both are wrinkled, and both are recessive, which means they are both the same as homozygous, and that is the reason why it is wrinkled in the phenotype. The alleles are the same, they are both recessive, and therefore they can be expressed in the phenotype. If there was another one, which would show you another round one, but it would be capital R, that would be both the point of the gene, both characteristics would be round dominant, and it is homozygous, which means that the phenotype will be round. Mendel's laws of inheritance. Grade 12, these you need to know by heart. You can see that I actually just snipped them straight out of the examination guideline. I have put the examination guideline page numbers there for your easy reference. You need to know these off by heart, and you also need to know how to apply them. So let's first go through them, and then later on in the upcoming examples and explanations, we'll actually put these into practice. The first one is the law of dominance. And it states, when two homozygous organisms with contrasting characteristics are crossed, so in other words, we are talking about contrasting characteristics as homozygous dominant and homozygous recessive. Okay, so they're saying that that's what they mean by the contrasting characteristics. Okay, so if we have a homozygous dominant individual and a homozygous recessive individual, all the individuals in the F1 generation will display the dominant trait. And what is the F1 generation? F1 refers to the filial generation, which is basically the offspring. Okay, so I'm going to say that again, because now I've explained to you a few things in there. So essentially what they're saying is that if you have two homozygous individuals that are different, so you have a homozygous dominant, for example, round, and you have a homozygous recessive, for example, wrinkled, then all the offspring, and you cross those, all the offspring in that generation will be heterozygous. Okay, and they will display the dominant characteristic, which in this example would be round. An individual that is heterozygous for a particular characteristic will have the dominant trait as the phenotype. That is what I just explained. Okay, so that in a heterozygous condition like the previous one, it would look like this. The dominant characteristic would be round, and the phenotype will also express that. You will find that on page 11 in the examination guideline. The next one is Mendel's principle of segregation. An organism possesses two factors which separate or segregate so that each gamete contains only one of these factors. Okay, so this is, remember I mentioned to you that when Gregor Mendel actually was carrying out these observations and experiments, he didn't realize that we are talking about characteristics and genes. He just understood that they were factors. So what he's saying over here is that all organisms in the diploid state, as we mentioned before, they are going to have these two factors, and they separate into each gamete, okay, during meiosis. So in this diploid organism, we're going to land up having two haploid gametes, right? And then this diploid organism is going to be showing two alleles. If we can represent an allele like this, one allele will go into the one gamete and the other one into the other gamete. Okay, so they are separated. That one you will find on page 12. The last one is Mendel's principle of independent assortment. The various factors controlling the different characteristics are separate entities, not influencing each other in any way and sorting themselves out independently during gamete formation. So when these gametes are actually formed, it's not like they are influencing each other about which way they're going to go. Okay, it's randomly. We see it earlier, this principle of independent assortment is also earlier when we're doing meiosis. When there's meiosis and they are going to be separating, or however the chromosomes are moving, it's not because of any other influence or influence on each other. They are moving independently. And this you will find also in your exam guideline on page 12. Like I said, please know these by heart. Right, now we're coming to the format of a genetic cross. I'm going to go through this slowly because it's very, very important. You'll almost always see one in every, basically in every paper two, you will be asked to do a genetic cross, and it's generally around 6 marks. Okay, so let's go through it. This is basically the recipe. We call this the recipe. Okay, it is the recipe, or it is your template, you could say, that when you are going to do this question, the template that I'm talking about is this part over here. Okay, if you just write down this in the correct order, you can get marks. Okay, but I'll go into that in more detail. Let's just go through this line by line. Okay, so first we notice this over here, say P1, that refers to the parent generation. And this P1, the parents obviously have a phenotype and a genotype. But what I want you to take note is that P1 phenotype is all in one line, right? Then the genotype is another line. Okay, let's just stop there for now. So you get your phenotype, which we explained is the physical trait. It is the phenotype. Let's use this example for tallness or shortness. Okay, so height basically. So whether they are, the characteristic, is height, and the phenotype would be either tall or short. Then the genotype is actually the makeup of the individual. That is the genotype. So the phenotype is dependent on what the genotype is. So this is where you're going to have letters. And both the phenotype and genotype, the question will guide you on what those are. That's where you get it from the question that is being asked. The next line after genotype is meiosis. Meiosis is suspended between the genotype line and the gamete line. Okay, it's important. This is a process. Meiosis is a process that occurs. And then in this process, we're going to have the formation of the gametes. The gametes is on a separate line. Okay. Before we go further, you took notice of these X's here these crosses in the center on X. This is indicating that it's very important that it is there in these 3 steps, and they are indicating that it's going to be the male. The male will be on the one side and the female on the other side. Okay, and also remember they are 2N, they are diploid, and they are showing that we are crossing, the cross is showing, they are crossing those 2 individuals. The genotype the same, and then the gametes, you would have, the genotype will then be separated. Okay, so to recap, the genotype is the one that's going to have the 2 alleles. Okay, so you're going to have 2 alleles over here at the genotype, and then due to the law of segregation, which we mentioned previously, that those alleles are going to separate, and they are going to be into the gametes. Okay, right, they're going to separate into the gametes. Then we are going to have a process called fertilization. This is another line, and in fertilization we are going to have the gametes, the alleles which are going to fuse. Okay, so let's back it up a little bit. We are going to have the 2 diploid organisms, as explained, with their phenotype and their genotype. Then they're going to go through a process called meiosis, where the gametes are going to be formed, and then those gametes are going to fuse. The sperm are going to fuse with the ova during a process called fertilization. So they are going to go from the one, so you'll have the one sperm, one possible gamete fusing with another one. These are going to show you at the end the possible genotypes. Okay, so these are the possible genotypes that can occur in this cross. So we have the one allele from the male and the one from the female. You'd get the first genotype, and then you will carry on taking the one from this side and one from the other side to get the next genotype. And then once you have the genotypes, you will express then what the phenotypes are. And F1 is once again referring to the offspring. Okay. And so in the offspring from these parents, we're going to have the possible genotypes, and then you have the phenotypes. This note over here is showing you that you get marks for indicating the P1 and the F1, as well as meiosis and fertilization. So that's why I said, even if you do not know what to fill in on this side later on, when you are actually doing examples of this and questions on this, at least if you fill out these in the correct places, then you would have, then you would at least get 2 marks. And then the other marks come from each line, okay? Those are where you'll get the other marks. Now, there's another way of doing this. In this instance, we have the format, another format. And the only thing that's different here is that we have a Punnett square. Okay, but I just want to go through it again. We have the P1 phenotype and the genotype, and then we have meiosis and the gametes will be formed. Then we'll go through fertilization, and instead of having the lines to cross, here we have a Punnett square. So the gametes that you get from here, you're going to put into these blocks, and these gametes will go into these blocks. Okay, and then you will determine the genotypes. When we do actual examples of this, I'll explain it in more detail. And then once again, you get the F1 phenotype and the genotype. The genotype you'll get from these blocks, and then the phenotype will be based on the genotype. There is a lovely explanation here as well, where you can get more information. And remember, like I mentioned before, remember that writing the P1 and the F1 and meiosis and fertilization in the correct sequence will give you 2 marks. Okay, so let's put this into practice. Right, but the first inheritance that we need to talk about is complete dominance. Okay, complete dominance is a genetic interaction where one allele of a gene suppresses the expression of an alternative allele in the F1 heterozygous. Example, capital B, small b, so that the phenotype is the same as that of the dominant allele. Okay, so in this instance, we are showing that we have, which we spoke about in Mendel's law as well, we have a homozygous dominant individual crossed with a homozygous recessive individual. The phenotype here is red, and over there it is white. But you can see in the F1, in the offspring, all of them are heterozygous, and they are all expressing the dominant allele. You can see they're all lovely and red. You can't see any white there, which means that the dominant allele, which is the red one, is suppressing or masking the white one. Okay, so that is Mendel's law of dominance. When we are discussing Mendel's law of we're explaining complete dominance, there are 4 types of crosses that can be carried out in complete dominance. Okay, we're going to go through the 4 of them. Number 1 is a homozygous dominant crossed with a homozygous recessive. Number 2, heterozygous crossed with the heterozygous. Number 3, homozygous dominant and heterozygous. And number 4, homozygous recessive and heterozygous. We will be using one general trait example, hair color and hair color. Okay, we will have brown and blonde. Right, so here we have the homozygous dominant versus the homozygous recessive, and we're going to go through it slowly. Okay, so once again, we have P1, which is the parent, and it's the phenotype, brown and blonde. Okay, so it's hair color, brown and blonde. And the genotype is going to be expressed, because they said it is homozygous dominant. Homozygous means that the 2 alleles must be the same. You can see here that they are the same, and it is dominant, which means it must have a capital letter. So they're both being expressed with a capital B. Homozygous recessive, also homozygous is telling us that they are the same. So they both are looking the same, yes, and they are the recessive, which means they're both going to be 2 small letter B's. Okay, then meiosis is going to occur. You can see meiosis is in a line on its own, and then we're going to have the formation of these gametes. Okay, so basically I always like to draw a line down the center of the genotype to show that this is now during meiosis that's going to divide. Okay, and then you're going to have this one gamete a possible gamete, another possible gamete, and on this side, possible gamete and another possible gamete. Okay? Then fertilization is going to occur where they're going to fuse, and in this example, we are using a Punnett square. So we're going to put the alleles into the Punnett square, the two capital B's over here, and the two small letter B's over there. Okay. Then the way they are crossed, you take this one B, this capital letter B, and this small letter B, and this is how you get that genotype. And then you take this capital letter B and this small letter B for this genotype, this capital letter B and small letter B for this one, and the same for the last one. Before we go any further, I just want to explain that each one of these little blocks, okay, each one of these possibles are basically going to give count 1 in 4. Okay, that's the ratio, 1 in 4, or as a percentage, each one is 25%. Okay, in the examples, in the following examples, we will actually go into that into more detail. This is just reminding you that the F1 is the first filial generation or the first offspring. And then once the genotype you're going to get from the Punnett square, as I mentioned before, and in this instance, it is capital B, small b, which is heterozygous. Okay, it's heterozygous, capital letter, small letter b, and it is 100% brown is the phenotype. Why is it brown? It's because brown is the capital letter B is coding for the brown allele, and it's the dominant allele, which is going to mask the recessive allele. Now, you may also be asked to work out the ratio or the percentage chance of the various phenotypes occurring. Okay, so in this instance, we used 100%. So we did the percentage, but you don't always get that. You have to actually read the questions carefully to see what is going to be asked. Heterozygous versus heterozygous, this is another cross, and this is between brown and brown. We have the P1 phenotype once again, and the phenotype, the physical characteristics, so what we are seeing on the outside would be brown, and then the genotype would be, which is expressed, the phenotype is dependent on what the genotype is saying, and in this instance, the alleles are different, they're alternate alleles. Okay, we have the capital B and the small letter b, heterozygous, the capital letter B and small letter b, another heterozygous. Okay, once again, we have meiosis. We're going to have the law of segregation, just reminding you over here, we have the law of segregation. The gametes are going to be formed, and then we have the possible gametes over here. Fertilization will occur. Once again, we plug in the gametes into the Punnett square. We cross them and join them, and we're going to be landing up with these genotypes. Now, when we are writing them down and we are referring to ratios, in this instance, we are going to say, okay, we're going to look at them, and you're going to count the ones that are the same. So here we have capital B, which is homozygous dominant, and there's no other one that looks like that, right? So it's 1. Then we have a colon, and then we do the next 2, which is capital B, small b. They are heterozygous. Here's 1, here's another one. So there are 2 of that genotype. Lastly, you have another colon, and then you've got homozygous recessive, small b, and that will be only 1. Now, looking at the phenotype, right? So here the genotype is showing us 1:2:1, but the phenotype, this is the genotypic ratio. But the phenotypic ratio, we're going to look at which ones are dominant, right? Brown is dominant. So in this instance, we're going to have, this one would be brown, this one would be brown, this one would be brown. And if they all count 25%, we're going to have 25 plus 25 plus 25, giving us 75% brown and 25% blonde. But if we look at the Punnett square, and I told you that they each count as 1 and we have 4, then we would say that the ratio would be 3 brown is to 1, referring to the blonde. So there the ratio would be 3:1. The next example is giving us homozygous dominant versus, with a heterozygous. Homozygous dominant in this instance would be the brown, small capital B, homozygous dominant, and heterozygous would be the alternate characteristics, the two different alleles, which would be a capital B and a small letter b. It is also brown because the dominant is masking the recessive. Then we'll have meiosis occurring again. The gametes will be divided. And I just want to mention something again as well. Remember we spoke earlier about 2N and 2N, okay? These organisms are both diploid, and then their genotype, these individuals are diploid, and then when meiosis occurs, then they're going to produce gametes which are haploid. The haploid gametes are going to be then N. Okay, so that's why we're saying they're separated and then they form into the different gametes. Those gametes go through fertilization. We plug them into the Punnett square. To remind you, capital B, which homozygous dominant. And then we have capital B, small letter b, heterozygous, capital B, homozygous dominant, capital B, small letter b, heterozygous. And here we have, this is the genotype is expressed over here, homozygous dominant, which would be 2:2, but ratios are always written in its simplest form, and that's where we get 1:1 from. And then they are 100% brown. They are all brown. You can see that the dominant allele is present there, and they are all brown. The last one we're going to be looking at, homozygous recessive and heterozygous. We have the P1 phenotype, which is the blonde and the brown. Once again, homozygous recessive. Now, the reason why it is blonde is because, remember, we said that when it is the two recessive alleles are going to be expressed in the phenotype. And in this instance, they're saying homozygous recessive, which is the small letter b, and then therefore the phenotype would be blonde. And the heterozygous is capital B, small letter b, and it would be brown. Meiosis will occur once again. Gametes will be formed. You plug them into the Punnett square, and you get these genotypes, which are 2 heterozygous and 2 homozygous recessive. Like I said, it must be in its simplest form. Therefore, the ratio is 1:1. And the phenotype would be, now we look at it, these two we have are heterozygous. The dominant allele is masking the recessive allele. Therefore, it would be brown. They each count for 25%. Therefore, it is 50% brown. And we look at the blonde, it is homozygous recessive, both carrying 25%, and we have then 50% blonde. Right, that is all from me. You can please wait for a few minutes. Nonhlanhla will be with you to continue the lesson. Don't go anywhere. Thank you.
Good afternoon, grade 12 learners. Welcome to the second session of today's lesson, which is life sciences, and it's on genetics, which is the first three topics as Vedika has mentioned. My name is Nonhlanhla Mtshali. Please don't forget to interact with us on the following number 060-667-5044. I'll repeat the number again. It's 060-667-5044. It is a WhatsApp number. Please do not send us any media, do not send voice notes, strictly typed messages. So I'm going to continue from where Vedika has left off. Before the break, we were talking about the different types of monohybrid crosses, the the different types of the complete dominance crosses, the 4 crosses that she mentioned that you must know, important that you know them when you go to your exams. So what we're going to do now, we're going to look at the question so that we understand why or the 4 that were mentioned before the break. So this question was taken from the 2018 paper. It's paper 2. Genetics is in paper 2, as it was mentioned earlier. If you look at the question, it says the flower color, which is purple or white, in a particular plant species is controlled by 2 alleles. Remember, everything is controlled by 2 alleles. And in this case, the alleles are capital letter D and small letter d. Then it goes on to say 4 crosses were carried out to determine which allele is dominant. 40 offsprings were produced in each cross. So we're looking at the 40 offsprings that were produced in each cross. The phenotypes of the parents and offsprings in a cross were recorded. So it is important that when you get a question, you read with understanding. Try to underline or highlight the things that you think are important to you, the things that you think are going to help you to respond to the answers. Then the question is, or the table says, there's 4 crosses. We were told that there's 4 crosses. There's parent 1, there's parent 2, and there's offsprings. And what is shown in the cross are the phenotype. Remember, the physical appearance. So when you look at the cross number 1, it's a purple flower crossed with a white flower, and we have 40 purple flowers. In the second cross, it's the purple flower or the parent with the purple flowers crossed with a parent with purple flowers, and the offsprings are 31 with purple flowers and 9 with white flowers. Number 3, we have a parent with white flowers crossed with parent number 2, who also has white flowers, and the offsprings are all white. And then number 4, we have a cross that has parent with purple flowers and is crossed with a parent with white flowers, and the offsprings are 21 purple and 19 white. Now, already in your mind, from what was mentioned earlier, remember you learned about the dominant and the recessive allele, the dominant being the one that expresses itself in a heterozygous condition, the recessive allele being the one that is not shown in a heterozygous condition. It will only appear if we're referring to a homozygous condition. If it's 2 recessive alleles, then that is going to show itself in the phenotype. But in the case of a heterozygous, always the dominant one is the one that shows itself. So the question number 1 says, state the dominant flower color. In the statement, we were not told which flower is dominant and which flower is not dominant. But then the question says, which flower or which flower color is dominant? So when we go back to the diagram, we can see that cross number 1, it's purple and white flowers. And all the 40 flowering plants that were produced had purple flowers. Whereas the rest, where we have purple and purple, we've got purple and white. Where we have white and white, we have white. Where there is purple and white, we have purple and white. So if we look at this, we can conclude and say, okay, cross number 1. In cross number 1, there were purple flowers crossed with white flowers, and all the offsprings are what? Are purple. So this tells us that this purple color is the dominant color, because in cross 1, there were purple and white flowers, but all the offsprings were purple. Now this brings us back to the first cross that was mentioned earlier under complete dominance, where if in there's a homozygous versus a homozygous condition, but one is dominant and the others are recessive, the dominant one will always show itself. So let's look at the answer for number 1. As you can see there, the dominant flower color is purple. Then number 2 says, use cross 1 to explain your answer. which I've already mentioned while I was explaining to you. To explain your answer in question 1, don't mind the 2.4.1, there's just an error there. To explain your answer in question 1. So when we go back, we are saying it is purple because if you look at cross number 1, a parent with purple flowers was crossed with a parent with white flowers, and the product were offsprings which all had purple flowers. There were no white flowers. So that is why we are saying purple is a dominant color. So let's look at the answer. It says when purple and white flowering plants are crossed, all offsprings have purple flowers, or there were no white flowers. Remember, you would say when purple and white flowering plants are crossed, all offsprings had purple flowers, or you would say when the purple and the white flowering plants are crossed, there were no white flowers, and it's 2 marks. Then the third question says state Mendel's law of segregation. Remember the 3 laws were explained earlier. When you look at Mendel's laws, there were 3 of them that were mentioned, and one of them was law of segregation. What does it say? It says that organisms they possess 2 factors or possess 2 alleles that separate or segregate during meiosis, so that each gamete only has one of these factors. That is Mendel's law. This you find exactly as it is from the exam guideline. Then the last question says, use a genetic cross to show how the crossing of 2 purple flowers plants can produce white offsprings in a cross. So we use a cross to show the crossing of 2 purple flowering plants. How can they produce white offsprings as in a cross? So when we look at the cross, remember, when you write across the formula, the recipe that was provided earlier, the parents, and then you show the phenotype and the genotype as shown there, and next on the same line where the phenotypes is written, the question says 2 purple flowers. So we have the purple and purple, this is the phenotype. Then the genotype, that's where we show the alleles. So in this case, because the question says how they produce the white flowers, so it definitely cannot be homozygous. So we cannot have capital letter D and capital letter D on both sides, because if that's the case, we are not going to get the white flowers. So in this case, it means we are talking about a heterozygous condition of purple crossed with another heterozygous condition of purple, which is capital letter D, small letter d, capital letter D, and small letter d. Then meiosis take place. Then these are our gametes. Remember, when you write the gametes, you must make sure that there's always a space in between so that you can clearly see that these are gametes. You are not talking about the genotype, because if you write them as shown above, you're going to lose the mark. It must always be a space, or you put the comma between the 2 letters. So we have your gametes listed there. Then fertilization takes place, which can be represented in the form of the cross, where you do a cross like this, or you do a Punnett square, a Punnett diagram. Most of the learners I know we prefer to do a Punnett square because it's easier than making those lines in a cross. Then the gametes are put there, capital letter D and small letter d, and then we have capital letter D and small letter d. Then with the cross, the result is the first filial generation, F1, the first offsprings. We have, the result will be the flowers that are purple, homozygous purple, because it's both capital letter D. There will also be a possibility of flowers which are heterozygous, a mixture. They have a dominant and a recessive allele, and also a combination of flowers that have small letter D and D, which is white. So the phenotype here is purple, 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 and white, and it is 3 purple is to 1. Remember, the question said, how is it possible that they produce white flowers? If you look at the memo here, there's an asterisk next to the white flower, because the question is asking about the white flowers. So when you write this cross, what is important is that this answer must be there. So even if you can write everything correct and you leave out this part, it means that you're going to get 5 out of 6, because you've left out the important part, which was the question, the possibility of having white flowers. Then the allocation of marks, as you can see, it is always 6 marks. they are allocated here, and then there's always these 2 marks that you get, even if you are not sure of what are you supposed to be crossing. But if you've got your formula written out correctly, guaranteed that you will get 2 marks for P1 and F1 and marks for fertilization and meiosis. Now moving on, we've already done complete dominance. Now we are moving on to incomplete and co-dominance. These are other examples of monohybrid crosses. Remember, monohybrid crosses, we're looking at one trait. One trait is involved in a cross. So with incomplete dominance and co-dominance, it is important that you still, or you understand the terms. Remember, we started with the terms. What is important is that you must know that we have the dominant allele. We also have a recessive allele. Then the dominant, we said it's the one that expresses itself in a phenotype, in a phenotype in a heterozygous condition. We said the recessive one does not show in a heterozygous condition unless the other allele is also recessive. So it will only show itself in a homozygous condition, but in a heterozygous condition, it is not going to show. So incomplete dominance, as the name states, it's a dominant that is incomplete, whereas this side we refer to co-dominance. So we're going to be looking at the red, white flowers, and on this side again, we're going to look at the red and white flowers. The reason why these are put side by side is so that you clearly see the differences between the two, because sometimes these two are confused. So we've put them side by side so that you can tell the difference. So let's look at the first one, incomplete dominance. So here we're looking at the red, the plant that produces red flowers crossed with a plant that produces white flowers. So when you look at the two, in this case, we're going to express the alleles as 2 capital letter R's for red, and we're going to express the alleles for white as 2 capital letter W's, because we are talking about incomplete dominance. So in an incomplete dominance, when you cross these two, when we refer to the question that we did earlier, the expectation will be that the flowering plants or the first filial generation will consist of the red flowers, white flowers. But in this case, when we talk about the incomplete dominance, a new phenotype is formed. So in an incomplete dominance, we get a condition where you cross 2 parents, let's say they are black and white, and a third phenotype shows up in the offsprings. It can be gray in the case of black and white. In this case, we've got a new phenotype. This is new and it is pink. So what does this tell us? It tells us that neither of these 2 colors are dominant. Red is not dominant over white, and white is not dominant over red. Therefore, a result is a new phenotype, and that phenotype is pink. So this is what we refer to as an incomplete dominance. So when we talk of incomplete dominance, we are saying neither one of the 2 alleles of a gene is dominant over the other, resulting in an intermediate phenotype in a heterozygous condition. So a new phenotype is formed. So this is very important that you know, and it's easier if we use the different letters. Under normal circumstances, when you talk about when we do complete dominant crosses, we were going to have a capital letter RR and small letter RR. But here we want to show that the dominance is incomplete. Therefore, we're going to use different letters, and your genotype here will be RW, and it's pink, and we represent it in capital letters because neither of the alleles is dominant over the other. Then when we look at co-dominance, this is co, meaning together. So already we can think that, okay, if it's co-dominance, it means then maybe both of them are expressed, or both of them are shown in the phenotype at the same time. So again, we're using the very same example of the red flowering plant crossed with a white flowering plant. So here the result is the flower that is a mixture of red and white. So what we see here is that both colors are expressed equally in the F1 generation. They are expressed equally. So they, like I said, codominants together. So they both show themselves in the phenotype. This is what we refer to as codominance. Remember, incomplete, we said neither of the colors or neither of the alleles are shown, whereas in the codominance both are expressed. So let's look at the definition. Codominance, we are saying both alleles of a gene are equally dominant, whereby both of them are expressed in the phenotype in a heterozygous condition. Remember, it is important that you put that in a heterozygous condition, because if the condition is not heterozygous, then you are not going to have the case. For example, if you do a cross where you cross now the red and white flowers together, you will get a condition where you find that some of the flowers will be red, and then you will get those that are showing codominance, red and white, and then you're also going to have to get flowers that are white. But if the two come together, they are expressed equally. So we get red and white flowers. We don't get pink flowers. So this is the difference between incomplete dominance and codominance. So let's look at the crosses. In the previous slide, we were looking at just the colors. Now we look at the crosses. Same as what we did when we're doing the 4 types of crosses under complete dominance. You will have your parents, your phenotype, and then we're looking at the red flowering plants crossed with the white flowering plants, and then the genotype will be expressed as 2 capital letter R's for red and 2 capital letter W's for white. Then we have meiosis, then the gametes expressed as capital letter R and capital letter W, and then we have fertilization. Then the cross takes place there. In complete dominance, it means that all the new flowers in the first filial generation are going to be pink. Hence, we have there 100% pink. Whereas in codominance, it's still the same. So don't be confused. Again, we'll be looking at red and white. Capital letter R, capital letter W, the gametes are expressed this way. And if you look at the genotype, it's the same. But because we know that this side is codominant and these flowers are red and white in color. So when we talk of codominance, in this case, we're just making an example of the flowering plant. You also see it in the chickens. If there's white and brown, I don't know if I could say brown or red, and then you find chickens that are brown with white spots or red with white spots. That is an example of codominance. Also, in humans, when you look at our blood types, we have blood type A, we have people who have blood type A, we have people who have blood type B, there are people with blood type AB, and there's people who have blood type O. So here, the fact that we have blood type A and B, it's an indication that these two are codominant. So if these two come together, they form AB, because neither A nor B, they are both dominant, therefore they express themselves equally in the blood type. But both of them at the same time, they are dominant over O. So O is the only one that is recessive. A and B are both dominant, therefore they express themselves equally. But you will do this, you'll learn about this more when we do the blood types. So let's look at one example. This one says in rabbits, before I look at the question, remember when you get a question, what is going to give you a hint that this is an incomplete or this is a codominant? There will be, sometimes when they give you a scenario, they will give you 3 colors, or they will tell you that the parents are brown and blue, and then they give you a 3rd color, which was not given as parents. So that should be an indication that they are referring to an incomplete dominance if a new color altogether appears, or a new trait appears which was not there before, or a new phenotype. Let me not say a trait, but a phenotype. Whereas with codominance, both of them will be shown in the offsprings. So this question says, in rabbits, fair color may be black, white, or gray. So we are looking at 3 phenotypes already here. Then the inheritance is controlled by 2 alleles, namely the black one, and the white one. So already from reading here, before we even go to the questions, we can tell that this is an example of incomplete dominance, because here we are given three colors. We are told that the fair color may be black, may be white, and it can either be gray. But what controls these colors? It's only two alleles, and those alleles can be black or what, or white. So now the question says, explain why fair color in rabbits is an example of inheritance with incomplete dominance. I've already mentioned that. Explain why fair color in rabbit is an example of inheritance with an incomplete dominance, because we are told that there's only two alleles that control a fair color, which is black and white, but we are also told that at the same time there is a color that is what? That is gray. But let's look at how the answer is given. Say that the answer says gray fair color is an intermediate phenotype, or it is a blend of black and white. This indicates that neither of the alleles is what is dominant. So neither black nor white is dominant. Instead, when they come together, they blend and they give us a color that is gray. Then the next question says, use a genetic cross to show the expected genotypes and phenotypes of the offspring when gray male mates with a black male. The question says to show the expected genotypes and phenotypes. So the compulsory mark here is going to be where on the genotypes and phenotypes. This is what we want to see. Although you're supposed to show all your work, you need to do the full cross for you to get to the genotypes and phenotypes and for you to get the full 6 marks. So when you look at an activity, this is what we look at. The question said, when you cross a gray and the black color, show the genotypes and the phenotype. So when you look at the first line, the phenotypes, we have the gray crossed with the black, and the gray is the combination of black and W. That's the genotype, and the black is just full homozygous BB. Then the gametes, as you can see from the gray one, the gametes, some of the gametes will contain B, and some of the gametes will contain W for white. Whereas in the black one, all the gametes will contain this allele, which is black, or this, the one that is black. And then fertilization is going to take place, and we've presented that in a form of a Punnett square. And then the cross between the B and the B gives us BB. Cross between the B and the B, it gives us BB. And between B and W, we have BW. And then between this and that, this is what we have. Then from here, we can see that the result is that some of the rabbits are going to have the black fair, and some of them are going to have the gray one. So if you look at the genotype, we have the rabbits that have the black fair, and we have the rabbits that have the gray fair. So the question said the genotypes and the phenotypes. So the first line shows the results of the genotype, and the second line shows the results in the phenotype. So some of the rabbits will be black and some of the rabbits will be white. So there's 50% chances that there will be black rabbits, and there's also 50% chance that there will be gray rabbits. Hence, we talk of the ratio, which is 1:1, because it's 2:2, therefore the ratio is 1:1. Then you have your marks there for P1, F1, meiosis and fertilization, and then the other 4 marks are going to come from the cross. Then the compulsory mark goes to the end. I hope that is clear. And then the next question is, we're still also on the complete and incomplete dominance. So the next question is an activity. It says, a farmer has an orchard of apple trees. Each apple tree produced expressed red and yellow color. So one apple showed red and yellow color, equally red and yellow apples. So red and yellow color were equally expressed in this one apple. Then to extend his apple orchard, the farmer collected seeds from red and yellow apples and grew them. Then when the new trees matured, he found that some of the trees produced red apples, others were yellow, and others, or the rest, were red, yellow. So we're looking at the apples that are shown red and yellow at the same time, and then the seed of these red and yellow were taken and they were sown and they grew, and then after that the trees that came out, some of them were just yellow, some of them were just red, red all over, and then some of them were still red and yellow. So let's look at the questions. The question says, use a genetic cross to explain his results in the first filial generation. So the question, when the trees were matured, he found that some were red, some were yellow, and some were red and yellow after crossing the red and yellow. So we would expect that red and yellow was crossed with red and yellow. So let's see what the answer looks like. So because he took the red and yellow apples, so the phenotype was the red and yellow apples that were taken, and then the genotype is RW because it's red yellow. Then meiosis takes place and the gametes are shown as some apples will have an allele for red and some will have an allele for yellow. So these are the gametes, and that's a mark. Then fertilization takes place, a cross is done here, and then the question said show the first filial generation. Show it says explain the results of the first generation. So this is what we are looking at. So the results were that some of the trees produced red apples, some of the trees produced yellow apples, and then some of them or most of them produced red and yellow apples. Because what we see here is that when the red allele and the red allele come together, they produce red apples. But when the red and the yellow allele come together, because they are codominant, remember we said codominant, so therefore they are equally expressed, then they will produce apples that are red yellow. And then when the yellow trait and the yellow trait come together during fertilization, they will produce yellow apples. So it's only when red and yellow comes together that both of them are dominant, therefore they express themselves equally in the phenotype. So this is how you do the cross for the first one, and that the marks are allocated as indicated. So the next question says, what proportion of apples in the F1 generation will be red yellow apples? What is the proportion of apples in the F1 generation will be red and yellow? Remember, earlier when Vedika was explaining, she mentioned that the ratio here is 1 because this is 1, and then we've got 2, so it's 2 and is to 1. So the ratio is 1:2:1, and when we add these, will give us 4. And so if we say 2, because the red yellow are 2, the ratio is 2. So if it's 2/4, 2/4 is 50% times 100. This is 50%. So the proportion that is given, it's 50% or it's half. So the proportion in all the trees that will be there, it means that there's 50% chances that most of the trees are going to give red and yellow apples. Then moving on, the next question says, the farmer saw that red yellow apples sell best. Name the phenotype of the trees that he should cross in future in order to ensure that any new trees will definitely produce red yellow. So after seeing that red and yellow sells a lot, so which trees should he cross to make sure that in future only red and yellow apples are produced? Then the answer is trees with red apples should be crossed with trees with yellow apples, because there, if it's red and yellow, we are guaranteed that red yellow apples are going to be formed, because red and yellow are codominant. So these are the pictures that we've shown. We're looking at the red apple crossed with a yellow apple, and these are the red yellow apples that we are talking about. You've seen these in the shops, so now you know why some of the apples are red and yellow. It's because the red and the yellow are codominant, therefore they express themselves equally in the phenotype. Then the next topic is on sex determination. Why are they males and females? How is it determined? How is the sex determined whether it's a male or a female. So we're gonna go back to meiosis. When you did meiosis, we look at the human, the karyotype. We looked at the karyotype, how the chromosomes are arranged, the chromosomes. So when we talk of humans, we say humans have 46 chromosomes, or we have 23 pairs. So in the 46, 23 comes from the mother and the 23 comes from the father. So we talk about 23 pairs of the chromosomes. So in the 23 pairs, the first 22, it is called the autosomes, and then the last pair, pair number 23, it is called the sex chromosomes or the gonosomes. So the sex chromosomes are the ones that determine the sex. They are the ones that determine whether the zygote is going to develop into a male or it is going to develop into a female. So when we look at the sex chromosomes, we say the males have X and Y chromosomes, and we say the females have X and S. So on the 23rd pair of chromosomes, we are saying males have X and Y and females and have X and X. This will be clearly explained in this diagram here. So here what we have is the karyotype of the female and the karyotype of the male. Then if the question would be, what is the difference between the two, or which between the two is the male and which one is the female? So if you look at the female, when you look at the 23rd pair, you should see two chromosomes that are the same. Same in appearance, same in size, same in everything. And if the last 23rd pair it consists of two chromosomes that are similar, then that means that that is a female. This karyotype is the one that represents the female. Whereas when you look at the male, the X and the chromosomes are not the same. There's one X chromosome because the X chromosome is usually bigger than the Y chromosome. Although we do not write it like this, I just want to make an example. The Y chromosome is smaller. So when you look at the karyotype on the last pair, the 23rd pair, if there's one big chromosome and one small chromosome, and then that should be an indication that this one it represents the male. It's the one that comes from a male. This cell comes from a male. So we are saying the first 22 pairs are called autosome, and the last pair is called the gonosome. So we have X and S in female, and we have X and Y in males. So what does this tell you before we even go on to the cross? Then this tells us that the males are the ones that are responsible for determining the sex of the child, because the females, if this cell has to undergo a meiosis, the result will be two cells or an ovum that only consist of an X. If these two separate and go to different cells, it will still be an X and a what? And an X. And an X and an X, they produce a female. Whereas in the male, we have this one big one, which is an X, and the small one, which is Y. So if these two were to separate during a meiosis, it means one sperm cell would have X chromosome and another sperm cell will have a what? A Y chromosome. And then it depends then on fertilization. So let's go to the next slide so that this can be clearly explained. So we are saying during meiosis, remember I said we're going back now to meiosis. During meiosis, we know that it is the type of division that results in the formation of the sex cells. So during meiosis, when the sperm cells are formed, the type of sperm cells that we have are, some of them, they will have 22 chromosomes plus the one X. Remember, I want to specify that the last one is an X. And then the other ones will have 22 and what? And the Y. So we have two types of sperm cells, those that are carrying the X chromosome and those that are carrying the Y chromosome. Whereas in females, as said earlier, it is the same. When meiosis in females results in the ovum, and all the ovums contain the X chromosome. There's no Y. Hence, we are saying it is the responsibility of this person to determine whether the the child is the male or the female. So let's look at the genetic cross now to show how sex determination takes place. So we'll start with all the crosses are done the same, regardless of what you are explaining. It's the same format that we explained earlier. So we start with the phenotype. In this case, we're looking at the male and the female that are crossed, and the genotype, we're looking at the sperm cells now, the formation of the sperm cells. We are saying the genotype is that we're saying in the karyotype of the males, they carry X and Y, whereas the females carry X and X. And then the formation of gametes after meiosis, we are saying sperm cells will contain X, some sperm cells will have Y. Whereas the females, the ovum that is released always have an X chromosome. Doesn't matter where we always carry X. Then during fertilization, now when fertilization takes place, chances are, okay, when X and X come together during fertilization, and then a girl child is born. Again, if X and X come together, a girl child is born. But if an X from the female crosses with a Y from the male, then a male child is formed. When an X from a female crosses with a Y, then the male child is formed. So we are saying that at the end, the genotype states that there will be 2 girls and 2 boys, or 2 females and 2 males. So what does this say? This says that there's always a 50% chance that the child can be a girl or a boy. It does not say that in a family, if a male and the female produce a child, it does not say that if they have 4 kids, then one will be a boy, one will be a girl, one will be a boy, and one will be a girl. No, it is saying in that one child that they are making, chances are 50% it can be a boy, or 50% it can be a girl. Because it does happen that in one family there's only 5 girls. Even if they continue to try, they will end up having 10 girls, because then it means each time fertilization takes place, the ovum of the female, which is X, is always fertilized by the sperm cell that contains X that comes from the male. So it lies with the male in terms of sex determination. So depending on which sperm reaches the egg, there is a 50% chance of the zygote being male and 50% chance of a zygote being female. I hope this is clear and you all understand. So let's look at an activity just to recap on what we've just spoken about. So the diagram below shows the karyotypes of 2 individuals. We've got individual P and individual Q. Okay, we're going to look at the questions, but let's just look at the diagram, which is what you should do. Before you look at the questions, look at the diagrams. What am I being given? What am I dealing with before I answer the question? So here we are given 2 karyotypes, individual P and individual Q. And we can already tell that this karyotype belongs to a human being, because there's 23 pairs of chromosomes on each. So if you look at individual P, and I said we focus on the 23rd pair. So if you look at individual P, we can already tell by looking at this that individual P is a male, because on the 23rd pair, there's 1 big chromosome and 1 small one. So definitely this is X and Y. Whereas if we look at individual P, both chromosomes are the same. They are equal in size. They look the same. So definitely individual Q is a female. So now let's look at the question. What term is given to the chromosomes numbered? Okay, it's not clear, but I assume numbered number 1 to number 22. So the chromosome numbered 1 to 22, this one. These ones we said they are called autosomes. Remember, that's what we said. And then we said the last pair, which is pair number 23, it is called the sex chromosome, or we call it the gonosome. So let's look at the question. Chromosomes number 1 to number 22, it is the autosomes, and the last one is the gonosome. So these are the 2 pairs. And then the next question says, state the gender of individual P. Remember, we've already analyzed the diagram. We know already we are not wasting time because we've done this. State the gender of P. We did say P is the male because we looked at the last pair and we discovered that these are not the same in size. They don't look alike. Therefore, we are saying this is the male. So here, give one observable reason to question 1.2, and the answer says it's the male. So we are correct in our analysis. It is the male, and one observable reason. This is very important. Not what you know, not what your teacher told you in class. What you can see from the diagram that is in front of you. So what do we see here? The observable reason is that chromosome number 23, the answer is given that it is non-homologous. They are not identical. They are not the same. On chromosome number 23, you can say one chromosome is big and the other one is small. Simply saying that, you've already earned your 23 marks because, your 2 marks. Specifying that at chromosome number 23, that's 1 mark. One chromosome is bigger and the other chromosome is small. That is your answer. So they are non-homologous, or you can say the karyotype P has chromosome X and Y on pair number 23. The karyotype has chromosomes X and Y on pair number 23. Then the question says now, Lindiwe has 2 sons. Lindiwe has 2 sons and she is now pregnant for the 3rd time. Use a genetic cross to show the percentage chance that this child could be a boy. If we go back to what I said, remember we said it does not mean that there will be 4 kids and 2 will be female and 2 will be male. We are saying that there's 50% chance each time. So let's see, use a genetic cross to show the percentage chance. We already know that the percentage chance is what? If you still remember, it is 50% all the time. It is always 50%. But let's look at the cross. So the cross is exactly as the one that we did earlier. Here is Lindiwe. Lindiwe always produces X and X. And here is Lindiwe's husband who will produce X and Y. And then depending on which male or on which sperm fertilizes the egg, we are saying there's always a 50% chance that the baby will be a male or 50% chance that the baby will be a female. And the question asked you to show the percentage chance of this child could be a boy. So the compulsory mark is where it's there, 50% male, because we are just focusing on what chances are there to say that this child could be a boy. So this is what we are looking at, 50% males. And you do a cross fully, you show your phenotype, your genotype, you show where meiosis is, you show where fertilization is, and you put your gametes in the form of a Punnett square. Then the phenotype is that 50% chance will be male or 50% chance that the child will be a female. And then this has brought us to the first section of genetics. I hope you understood, and I hope you did interact with us through the questions. And remember, COVID is still with us. As much as we were told to remove our mask, you still wear your mask in classroom, you social distance, you wash your hands. And if you have more questions, please let your teachers know. If there are things that you do not understand, they will let us know, and whatever issues that you still have will be addressed in the next session, which is on the 20th. I think our next session for genetics is on the 20th. So please tune in on the 20th, same time, 2 to 3:30. And the number for WhatsApp interaction has not changed. It is still going to be 060 667 5044. Thank you very much.